All right. I hope everyone had a nice lunch, and I guarantee you we will not have issues with postprandial coma because this is going to be the most, I'm a little bit biased here, but I think this is going to be the most exciting part of the family conference. So we've been talking about basic science and sort of fundamental concepts, but at the end of the day, I think most affected individuals and family want to know about treatment or a cure. And that's what we're going to be addressing over the next couple hours. And a lot of people have heard of gene therapy and gene editing, and people have various levels of understanding what that means. And RR1 is sort of, like most rare diseases, has some unusual features. And we're very lucky to have Dr. Isabelle Marti here today. Dr. Marti is a leading expert with over 30 years of experience studying RYR1. She specializes in developing therapies in RYR1-related diseases, including gene therapy. She's actively involved in French patient organizations, including AFM Telethon. She's a valued member of our scientific advisory board. And today, Dr. Marti will share the latest advances in gene therapy research, what this means for neuromuscular conditions, and what the future may hold for RYR1. I will tell you, anytime I start thinking about RYR1 and gene therapy, I go to Dr. Marti's papers and her publications, or I will email her directly. She is the go-to authority on this topic. She presented on this topic at our research workshop. I don't think you're going to find a better authority on this topic, so we are really lucky to have her today. So Dr. Marti, please welcome to the stage, and thank you very much.
Thank you, Michael. Thank you for the invitation, and I am really happy to be here today to present you the progress in treated neuromuscular disease and the future hope of gene therapy for RYR1-related myopathy. So just as an introduction, this is the place where I live in the French Alps. And I will start with very basic concepts, so I apologize for those of you who are familiar with all these concepts, and probably you heard already about it before. So I will start very slowly, just to remind you how a skeletal muscle works. So probably you all know that the muscle is under the control of motoneurons that come from the spinal cord, represented here, and the spinal cord transmits the order to contraction to the muscle. So the nerve influx, which is this order, travels along the motor neuron and then will stimulate the muscle. When this nerve influx reaches the muscle, it will induce the calcium, which is contained in a small tank, into the muscle. It will induce this calcium to release into the muscle, and this calcium will induce the muscle contraction. Muscle disease can have very different origin, and the defect can occur anywhere on the way to contraction, either on the spinal, on the motor neuron, on the neuromuscular junction, or anywhere on the muscle. Our focus today is exclusively the disease resulting from alteration in calcium release. So you all know that RYR1 is a main player between muscle stimulation and muscle contraction. And this protein, RYR1, is a calcium channel, which is at the center of a so-called calcium release complex, with a number of associated proteins these are partners which helps the regulation of calcium release. And when RYR1 is stimulated, the calcium is released in the muscle cell, and as I told you before, this will induce the muscle contraction. What are the consequences of RYR1 mutation? They all lead to reduction in calcium release. They can either have an effect on the calcium channel function with what we call a gain of function, which means that in this situation, the calcium is released permanently and therefore the calcium store is empty, or a loss of function. In this case, the calcium is not released, but there is also another situation. It's the reduction in protein amount, which means that there is no more channel and the calcium is not released. if I compare the AirWire1 and the calcium store to a faucet on a water tank, it's not like Bob this morning, but it's a little bit similar. So the AirWire1 is the faucet, and the calcium store is a water tank. In this case, in gain of function mutation, the faucet is leaky. In loss of function mutation, the faucet is blocked. And in a reduction in protein amount, there is no more faucet. And it is of major importance to understand the precise mechanism in order to identify the most suited therapeutic approach. Now, a brief overview of genetics, so very simple. You probably know that each cell of the body has a nucleus, represented here, which contains the DNA, and the DNA is the memory of the cell. The information contained in the DNA will be transformed into a protein, and this is the protein which are the worker of the cells. The DNA form genes, and these genes assemble to form chromosomes. Each individual has 20 pairs of chromosomes, therefore 2 copies of each gene, one coming from each parent. And when there is a genetic disease, there is a mutation in one or in the 2 copies of the gene, and this is transmitted by one or by the 2 parents. The therapeutic development, so you know that when there is a mutation in the AirWire1 gene, this will result in an abnormal AirWire1 protein, and this is also causing an abnormal muscle function. The physical therapy aims directly at restoring or improving the abnormal function. The pharmacological therapy will target either the abnormal protein or the abnormal function, and the gene therapy will be just above all and try to correct directly the mutation in the AirWire1 gene. What is the pipeline for therapeutic development? So the first thing is to identify the problem due to a mutation. Then it's a demonstration, the proof of concept of a therapeutic strategy, because we have sometimes a very clever idea. We have to demonstrate this, the benefit of the therapeutic strategy in a cell model. And then we develop animal model, and we test this therapeutic strategy in animal models. So the animal model can be small animal model, mouse, but it can also be larger animal model like dogs. And if everything goes well, then it will go up to a clinical trial. And if everything goes well, then a new therapy is on the market. So let's have a closer look at gene therapy. So gene therapy, of course, it's the gold standard because it can cure the initial DNA defect. But few issues have to be solved first. The muscles are spread in the whole body, and all the muscles have to be targeted to be cured, and especially the respiratory muscle, which are the most difficult one to reach. Therefore, the gene therapy molecule will be delivered by systemic injection for delivery by the blood circulation. So the bloodstream will allow the delivery to all the muscle in all the cells, in all the nuclei. What will be injected for DNA therapy? The most powerful tool today are viral vector. So this viral vector has been modified. They were initially designed from the HIV virus responsible for AIDS, and all the dangerous parts have been suppressed. So these viral vectors have been engineered to suppress all these dangerous parts. They are highly efficient because they can enter into all the cells and transfer DNA, but they have limited capacity. They cannot transfer very large things, and they can result in possible immune problem because our body is dedicated to fight against the viruses. So from theory to practice, has gene therapy with viral vector ever been applied to a neuromuscular disease? Yes, this has been the case for myotubular myopathy, also called X-linked centronuclear myopathy. And this myopathy is due to the lack of a protein called MTM1, and the first demonstration of the benefit of therapy has been obtained in a mouse model and then in a dog model of the disease. This has been a long-term collaboration between many international teams in France, in US, in Canada, and the first trial has been done with the injection of a viral vector encoding MTM1 gene, and this has been shown to reverse the disease. So I will show you the amazing result obtained in muscles in a dog model.
We're able to measure strength in dogs.
I don't know if you can hear.
In fact, even at about 4 months of age, there's some weeks they can no longer stand.
So you can see here affected dog.
Just with the test dose, we saw within just 30 weeks.
And the dog has been treated. You can see that he behaves much better. He can play.
6 weeks, the strength in a very, very weak muscle was entirely normal. That was just absolutely shocking to us. In fact, we thought something had gone terribly wrong with the equipment. We just couldn't believe it. But this has been replicated now enough times.
And 10 months after treatment, you can see that these dogs are playing. They behave completely like normal dog.
Very bad, fatal disease.
So this has been just amazing, and this result lead people to develop a clinical trial for children affected with the very same deficiency. So I will show you another movie. It's the story of Jules, a small French boy who received gene therapy vector encoding this MTM1 gene. So I apologize, the movie is in French, but you have English subtitle, and maybe some of you can understand French in the room. So hopefully it will work. Sorry.
À sa naissance, on ne savait pas ce qu'il avait, forcément, c'était une maladie rare. D'ailleurs, personne ne savait même nous dire si c'était une maladie. Très vite, ils ont compris qu'il y avait quelque chose qui n'allait pas. Et donc, ils ont fait les recherches aussitôt. Et après 2 mois, ils ont su nous dire que Jules était atteint de myopathe tubulaire. Maladie des muscles, maladie généralisée des muscles, qui l'empêche de se développer correctement. Enfin, il était hypotonique au complet, il bougeait très peu, il respirait avec une machine, il mangeait avec un appareil. Enfin, toute sa vie tenait grâce à des appareils. Quand on nous a annoncé la maladie de Jules, on nous a annoncé durée de vie moyenne, quelque chose comme 1 an. On a pris ça comme un grand coup de massue. On a pris contact avec l'AFM Téléthon, pour savoir ce qu'il en était, ce qui en découlait de la maladie, et qu'il y avait un espoir de guérison, quelque chose pour nous, quoi. Et on nous a renvoyés vers Emotion. Tu tiens
Je suis juste là, c'est bon. Je ne vais pas t'embêter plus que ça.
Jules a eu une injection, donc un traitement. C'était un essai thérapeutique. Et nous, on avait espoir que Jules récupère certaines facultés. Moi, j'avais envie qu'il soit autonome au niveau des machines. Moins on a de machines, forcément, plus la vie est facile, quoi.
Presque 15 jours après l'injection, on voyait déjà des effets. Donc ça a été très rapide, on a eu des effets très rapidement. On est 6 mois post-injection. On n'aurait jamais... Enfin, nous, on s'était fixé chacun des petits objectifs personnels. L'évolution, elle est juste exceptionnelle.
Il sort, il fait du vélo, c'est déjà pris des gadins, mais bon, ça, voilà, ça fait partie du truc, quoi. Et puis maintenant, il est plus costaud, et puis maintenant, il a à peu près trouvé le truc avec son tricycle. Il est de plus en plus autonome. En fait, il a complètement dépassé nos espoirs, quoi. Il est devenu autonome à tout point. Enfin, il suffit de le voir aujourd'hui, il marche, il monte les escaliers tout seul, il rentre dans son lit tout seul, il est même capable de se coucher tout seul. Il a récupéré une bonne partie de son autonomie.
est au-delà de ce qu'on s'était fixé.
Je vais vous présenter quelque chose. Ça va pas la tête? Oh la binette. Là. Alors, oui. Reviens là, toi. Oh, ça va bien. Le bateau va démarrer dans 3 minutes. Moi, je vais là-bas pour leur chercher des cailloux, des cailloux. Et je vais faire une maison. Je vais fabriquer un château de sable qui est géant jusqu'au ciel. Alors ça.
So this is just an amazing story, and nevertheless, I have to mention that the treatment now is stopped because a few deaths occur in some of these children. The ones that have been treated, they are still doing very well. So this is very full of hope. And the question is to know if such a gene therapy can be applied to RYR1. Unfortunately, no, because there are some major differences between the MTM1 gene and the RYR1 gene. The first one being the size of the gene. So it's not at the scale, but you can see that the RYR1 gene is so huge that it is too large for a viral vector. And no viral vector will be able to fit the RYR1 gene. And it's not possible to re-express the RYR1 gene. In addition, the type of mutation is not the same. For MTM1, there is no more expression, but in patients with dysfunctional RYR1, which are dominant mutation, with a leaky faucet, if I take the analogy I presented you before, there will be no benefit of a re-expression of RYR1. So now we are trying to look at what is done for large gene, because we understood that the problem of the size of the gene is something that has to be solved. So we are looking what is done for Duchenne muscular dystrophy, which is related to mutation in the dystrophin gene. So what is dystrophin? It's a very large protein. It has about the same size as RYR1, and it has two important parts at each end, at each extremity, with repeated modular domain in the middle, which seems to be less important. What happens when there is a mutation in this gene, in this protein, in fact, there is no more protein production. So what has been done for dystrophin, because it is too large for a viral vector, the first strategy is exon skipping. So it consists in introducing in the cell small DNA segment to induce the skipping of the region which is mutated and to induce the production of a shortened but functional protein. And the second strategy is to express a mini or microdystrophin with a viral vector, which means to induce the expression of a shortened protein which will fit into a viral vector. These are the current proposed treatments, and the ongoing long-term benefits are being studied currently. And the third option is to express dystrophin in multiple fragments, which are multiple vectors, that will reassemble together to form a full-length protein. This is, for the moment, a research project. This is not in trial in patient. So can this DMD strategy be applied to RYR1? So exon skipping, no, as we heard this morning, it's not possible to use exon skipping because no part of RYR1 is dispensable. Is it possible to express a mini or a micro RYR1 with a viral vector? No, because no mini and functional RYR1 has ever been identified. Is it possible to express RYR1 in multiple fragments within multiple vectors that will reassemble together? This is something that has to be tested, it will require at least 4 viral vectors and an increased efficiency, and you will probably heard about it later this afternoon. So we are left with gene editing. So what is gene editing? It's the direct correction of the mutation on the DNA. This is a powerful but very recent technology. The 2 women who discovered this strategy, Jennifer Doudna and Emmanuelle Charpentier, win the Nobel Prize in 2020 for CRISPR-Cas9. So it's very recent, and it is mainly used in research for the moment. Improvements are required, but a first patient has been treated here in the Children's Hospital of Philadelphia for a very rare and severe genetic disease, liver genetic disease. So how does genetic editing work? So the gene editing, which means the gene correction, is performed at the DNA level using Cas9 nuclease and double-strand DNA cleavage. So don't be afraid by the word, I will try to explain. So the nuclease, Cas9, means that it is something that can cut the DNA, a guide RNA, which will guide the cleavage of the DNA, and a DNA repair system. So the nuclei and the guide assemble together, and then they travel along the DNA, and when they found a very specific sequence, which is called the PAM, the DNA, the nuclease and the PAM and the guide stop on this sequence. And if there is complementarity between the guide and the DNA, then the nuclease will cut the DNA. And the cell does not like double-strand break, does not like to have its DNA cut. And the cells have multiple mechanisms to repair this break. Either some perfect repair, but this is not working in muscle. And in muscle, what is working is what we call imperfect repair. And most of the time, this results in deletion of the gene which has been repaired in such a way. So we have to take the opportunity of this imperfect mechanism to perform gene correction. So how do we use it in my team for AYA1-related myopathy? So we have observed in the past that 2 copies of AYA1 are present in each cell, but only 1 copy is enough to perform the correct function. So when we have 2 copies and 1 is mutated, which is the case of a dominant mutation, we try to erase the mutant copy of the gene and to preserve the normal one. So this is represented here and much better here. So when you have the 2 copies of the AYA1 gene, 1 with a mutation, you have a very small calcium release because it is a dominant mutation. Using gene editing, we try to delete this mutant copy and keep just the normal one, and this will result in a normal calcium release. So this is what we have done, and we have shown that this is very powerful in patient cells, and we use viruses to introduce the CRISPR-Cas9 tool in all the muscles in the patient cells, and now we are working on a proof of concept in a mouse model. The second strategy is prime editing. It's the surgical correction of a mutation with insertion of the chosen DNA segment in place of the mutant one. And as for CRISPR-Cas9 gene editing, it needs the integration into the cells of the tool to perform the correction. And therefore, we have the same issue with the efficiency of the delivery. The precise edition using prime editing, in addition, should be adapted to each patient and to each mutation. Nevertheless, a first successful application has been obtained recently by a Canadian team. So what are the next challenges for gene editing and gene therapy? The proof of concept of few gene therapy strategies have been provided in a muscle cell culture for AYA1-related myopathy. So now the demonstration in an animal model has to be performed. So it will be first in a mouse model because we have no other model. And we need absolutely to improve the delivery and the efficiency. First, to reach all the muscle, and second, to reach only the muscle. And therefore, we have a lot of work to develop new vector. And therefore, this is common to all the myopathies. So many teams are working on the vectorology. And the goal is to reduce the doses that we have to inject to a whole organism. And this is also in order to ease the vector production, to reduce the cost and to reduce the amount of virus required, because for the moment, each dose for one patient is over $1 million. And we have also to limit the side effect. So if I summarize, we are on the way to gene therapy for AYA1-related disorders. It can be done in muscle cells, and now we have to work to improve it and show the demonstration in the animal model and in human. And the improvement is needed for the delivery and the efficiency and for reduction of the cost. So I would like to finish by something which is full of hope. This is the visit of Antoine, a French boy affected with a very severe form of AYA1-related disease, who visited our lab a few years ago. And at the end of his visit, because he has never come into the mountain, he decided to discover the mountain using paragliding. So this is just to say that you have to keep hope and that something which looks like impossible can be possible. And also, I would like to thank our major funder, which is AFM Téléthon, the French Muscular Dystrophy Association, which has supported my work for more than 30 years now. Thank you for your attention.
Thank you, Dr. Marti. Yeah, hang gliding is tonight. I know that wasn't on the schedule, but any questions, comments from the audience? Mary Beth, could you go use the microphone?
I had two questions, if possible. Thank you so much for the important work you're doing and for coming here to see us. First, I would like to ask about mutation-specific versus more universal approaches. As we know, there's a wide array of RY/R1 mutations, and your talk focused a lot on the specific mutation-specific editing. Is anyone looking at a more generalized approach where perhaps you could introduce like a protective mutation, stabilize the channel somehow, maybe? That's one question, just curious.
In fact, it is one of our goals in my team. We are trying to develop a strategy which is not dependent on the mutation. Because of the cost of the development of the treatment, we try to develop the strategy to erase the mutant allele. I forgot to mention whatever the mutation is. So the strategies that we have developed is with the very same tool, we can treat 20% of the patient with a dominant mutation, whatever the mutation is. So this is something which could be very powerful and very depersonalized medicine, because too much personalization is not good. We cannot afford a treatment for just one patient.
Yeah, thank you. My second question was, how do you think gene therapy might integrate with or support some of the emerging therapies, which you may or may not be aware of, the RICALs and the small molecules? Do you think that there could be some synergy there?
Of course, it's not a competition. I mean, all the strategies are good, and the gene therapy, it's not for tomorrow. I think you understood that for the moment we are just at the cell level. We need to go to the animal levels and to the clinical trial, and even the MTM1 gene therapy, which is very, very efficient, is stopped nowadays because of patient death and because of economical issue, because no company wish to develop a therapy in which the patient can possibly die. So in fact, The other strategies are also very important, very interesting, and if it's not a definitive cure, it will help. Whatever the strategy, it will help, and we have to combine every kind of approach.
Thank you.
Any other questions? Oh, here we go. Lauren?
Related to what you talked about with like the MTM therapy trial being put on pause to deaths, and then I was remembering like a week or two in the news here, I guess it was for neuromuscular dystrophy, another trial for gene therapy being put on pause due to deaths. With stuff like that, obviously I don't know if they're using kind of a similar delivery system, but are there common themes when you do see complications in gene therapy trials of what's going on with those patients that's obviously causing, medical issues or even a death?
I think, in fact, that in most of the case, it's because we haven't done enough basic research, and we wanted to go directly to the treatment, we wanted to go to the clinical trial, but for MTM1, for example, what has been observed is a death because of liver issue, and now the basic researchers are coming back to study the animal model, to study the patient, and they discover something that they haven't seen before, that the patient have liver issue, and this is the reason why the gene therapy lead to the death of the patient. In the Duchenne muscular dystrophy, it was another issue. The problem was that the vectors they inject has some part which were highly immunogenic, which means that the body has never seen this part of the protein before, and then there is a huge immune response against the protein. So each time we didn't do enough basic research to understand all the different aspects of the disease. This will lead to a failure of the clinical trial. So it's very important to understand that everything is complementary, and we cannot make the economy of basic research.
Any other questions? Right here?
Can you take the...
You just said there was risks to the liver. Reason why I'm asking is because what I've learned, my son has RYR1, but he also has the ketotic hypoglycemia. So I've had to learn how the function of the body works between the liver and the muscle. So are you seeing with these trials it affect the liver? Is it because of the stores with the sugar regulation in the body?
For MTM1, I'm not really familiar with MTM1, but I understood that there was an alteration of liver function in the patient, which has not been observed correctly before and described before and analyzed before. So I don't know why exactly, but it was involved in the liver function. RYR1 is not involved in the liver function, so there should not be any correlation, but as you said, glucose is important, is produced in the liver, it is important for the muscle function, so yes, you can have some interaction, and this is also the reason why we are testing the therapy in an animal model, because you cannot just test a therapy in cells, because you have communication between the different organs, and this is what is important for the success of a therapy. So this is the reason why we have to test it in an animal model, and in an animal model as close as possible as to human. So usually we use primates, but this is, I don't know how it is in US, but in France it's just horrible. So I think the only place where you can do primate test very easily, it's China. So maybe in the future everything will be done in China, I don't know.
Any other questions or comments? Thank you, Dr. Metti. That was fantastic.
Thank you.
Thank you.